And before we get started, I would just like to take a moment to thank the hardworking volunteer committees whose vision has made this possible. First, thank you, Dr. Cheryl Boyer of Kansas State University, for hosting and for sharing your expertise on digital communications. It's really been a sincere pleasure for the last 4 months Zooming with you on a near weekly basis. I'd like to thank the Student Competition Planning Committee that was able to get organized very quickly because we didn't really even think this was going to be possible until July. And that includes Dr. Mac Thetford, the committee chair, Dr. Cheryl Boyer, Dr. Chris Marble, Executive Secretary Donna Foster, and Digital Communications Manager Laura Barth. Thank you so much for your hard work and dedication in bringing this all to life.
So our first speaker today is Isha Powdall. She is a master's student from Tennessee State University in McMinnville, Tennessee. She completed her undergraduate degree from Tribhuvan University in Nepal. Currently, she's working with Dr. Anthony Witcher at the Otis Boyd Nursery Research Center, and her work focuses on weed management in the propagation environment. And her goal is to become a leading scientist in an academic research laboratory or in industry. The title of her paper was Mulch Depth Effect on Rooting Stem Cuttings and Weed Control During Propagation. And the authors on that paper are Isha Powdall and Anthony Witcher. So if you would, please.
My research topic is on mulch depth effect on rooting stem cuttings and weed control during propagation. Weeds are the major problem in propagation. Propagation is mostly done in small containers, due to which it creates more competition for nutrient, light, and water between the crop and the weed species. Also, propagation requires frequent irrigation, due to which it creates favorable environment for weed to germinate and grow. Controlling these weeds in propagation is very challenging. Growers normally adopt hand weeding for controlling weeds in propagation, but it is very costly, labor intensive, time consuming, and suppress growth of cutting through mechanical disruption. Also, pre-emergence herbicides can be used as alternative to controlling weeds in propagation, but they are not labeled for used in non-rooted cuttings and inside enclosed structures. In this situation, mulches can be used as alternative to hand weeding and pre-emergence herbicides. Mulches are being widely used in nursery crop production for controlling weeds with great success, but there are very limited research on their use during propagation. Hence, a study was initiated with an objective to determine the effect of different mulches and application depth on rooting of stem cuttings and weed control in propagation. A rooting experiment was done using 6.6 centimeter diameter container with pine bark as a substrate. 4 different mulches, namely vermiculite, rice hulls, pine pellets, and paper pellets were used at 0.5 and 1 inch depth. Softwood cuttings of hydrangea were stuck in each container after application of all the mulches. All the containers were completely randomized and placed under safe and intermittent mist. Data were collected on rooting percentage, root dry weight, shoot dry weight, and digital root analysis was done to get root length and root volume. And data were analyzed using SAS. And for weed efficacy experiment, we use same size container and substrate. All 4 mulches were applied at 0.5 and 1 inch depth. After application of all the mulches, seeds of 2 weed species, crabgrass and bettergrass, were sown. Containers were completely randomized within species and kept under shade and intermittent mist. Data were collected on weed count at 2, 4, and 6 weeks after sowing, and shoot fresh weight was collected at 6 weeks after sowing, and data were analyzed using SAS. And from all of this, we got satisfactory result. For rooting experiment, rooting percentage was 100% for all treatment except rice hulls, which was 90%, and paper pellets, which was 95%, applied at 1 inch depth. Root dry weight was similar to control for all the mulches, and even total root length were similar to the control for all the mulches. Wherever root volume was lower for paper pellets at 1 inch depth. And for weed efficacy experiment, crabgrass seedling count were similar for all treatment compared to non-treated control, and betagrass seedling count was lower for pine pellets at both mulch depth. Pine pellets and paper pellets suppress shoot fresh weight of crabgrass and betagrass at both the mulch depth. From this study, we found out that mulches have good potential for controlling weeds in propagation. And from our study, we concluded pine pellets and paper pellets were most effective for controlling weeds in propagation. Even though the weeds germinated in pine pellets and paper pellets, they couldn't grow well. But paper pellets resulted in slight reduction in root growth of hydrangea, but as compared to high risk of injury due to herbicide use and high cost of labor associated with hand weeding, it is very acceptable. And we are further doing our research on determining the physical and chemical property of these mulches to find out which factor is contributing for the weed suppression. And also we are trying to find what will be the effect of these mulches on crop after they are transplanted. Thank you so much.
Thank you, Isha. We appreciate your presentation. Our next speaker is going to be Ping Yu. Ping is a PhD candidate at in the Department of Horticulture Sciences at Texas A&M University. She completed her master's degree at the Nanjing Forestry University in China and started her PhD program with Dr. Meng Gu at Texas A&M in 2017. Her research focuses on using biochar to increase environmental friendliness for potted plant production and disease suppression. Her professional goal is to become a professor in the area of extension after graduation. The title of her paper was Effects of Gibberellic Acid and Cold Stratification on Sparkleberry, Vaccinium arboreum, germination under different collecting times. The authors on her paper were Ping Yu, Lin Li, Quanxing Li, and Meng Gu.
Dr. Zephyr, how many minutes do I have to present?
5.
Okay, yeah, just make sure.
Yep.
Okay.
Can you see me?
Yeah.
Okay, I'll change colors if you, in the last 30 seconds.
All right,
But
thank you.
you guys are doing great.
Thank you so much. Can you guys see my slides?
Yes.
Okay, perfect. Uh-oh. Okay.
Can you swap screens?
Sorry about that. I have like 2 monitors, so it just switched to another one. So I'm gonna redo it, the whole thing again, and try if that's gonna work. Share. And yes, there we go.
There you go.
Okay. Thank you so much for the introduction. So my name is Ping Yu, and I'm currently a PhD student in Texas A&M University working with Dr. Meng Gu. Last year, I presented my biochar work at IPPS. So this year, we're gonna talk something different. Today, I'm going to talk the effect of gibberellic acid in cold stratification on sparkberry germination under different connection times. So first of all, why we care about sparkberry? If we're going to talk about sparkberry, we have to talk about blueberry first. So as you can see, all those big numbers showed here that indicate an increased trend for blueberry demand. But one of the big challenges for blueberries that we are facing right now is that the blueberry requires specific soil, such as lower pH and high amount of organic matter. In contrast, sparkberry has all the traits or all the desirable traits that we want the blueberry to have, such as low amount of organic matter and singular architecture. So by grafting, we can use sparkberry as a rootstock for blueberry and to make the blueberry to have it all. However, as we all learned this year, not all things can go smoothly as we want. So for sparkberry, it has its own limitations. The biggest one that we're facing right now is that it is very hard to root by cutting, either by softwood cutting or hardwood cutting. And for now, the highest recorded germination rate by state for sparkberry was 43. So if we want to use it as a rootstock for blueberry, we have to produce it in a large scale. So in this study, we explored the potential positive effects of GA3 and co-certification on sparkberry germination rate to further benefit blueberry industry. This is how we did our experiment. We collected seeds and treated with GA3 and co-certification and measured the emergency percentage for sparkberry. Here is our results. So we have each panel we have 4 panels in this figure, and each panel represents the stratification period for 0, 3, 6, and 9 weeks. And in each panel, we have 3 different lines that represents different GA concentration. That was 0, 500, and 1,000 milligram per liter. So as you can see, for states haven't been stratified, had lower emergency percentage in comparison to those who had co-certification, while the highest emergency percentage for November state here was reached in here, co-certified for 9 weeks, and GA applied with 500 milligram per liter. And for November states, similarly, the states without co-certification had a lower emergency percentage in comparison to those who had. And the highest germination rate or emergency percentage was reached only for 3 weeks of co-certification with a higher GA3 concentration, which was 1,000 milligram per liter. So the results or the conclusion for this study was that the GA3 treatment and the co-certification could be used together to improve sparkberry germination. And for November states, a 70% of germination rate could be reached with 500 milligram per liter GA and 9 weeks of co-certification. And for December states, around 60% of the germination rate could be reached with 1,000 milligram per liter GA and 3 weeks of co-certification. So what does that mean? We wanted to know what does that mean. So this study increased the sparkberry germination from 43 to more than 70%. That means we can produce it in a large scale and can use it as a rootstock. And that means we can significantly reduce the blueberry yield loss caused by the mechanic harvesting, and we can create more than $100 million for blueberry industry and reduce a lot on the organic matter costs in blueberry production right now. So we would like to thank all those people who helped us. And with that, I would like to take any questions. Thank you.
Thank you. We're going to do a question and answer session immediately following our next 2 speakers. So be sure to write down your questions, and we'll have those in a few moments.
All
So
right.
our next, go ahead. Okay.
No. You got it. You got it.
Our next 2 presenters are going to have the challenge of presenting their work in only 2 minutes. For those of you who have heard of a Scholars Ignite type presentation, or tell me what you're doing for your research while we ride the elevator, this is the challenge. So the first of our speakers will be Runqi Zi from Texas A&M University. Runqi received his bachelor's degree in landscape architecture from Fujian Agriculture and Forestry University in Wuxia, China. Runqi came to Texas A&M University for his master's degree in plant breeding in the Department of Soil and Crop Sciences. After completion of his master's study, he joined the Department of Horticulture Science to pursue a doctorate in horticulture. His current research focuses on developing strategies for controlling and managing crepe myrtle bark scale and introduced pest species in the United States. His professional goal is to work in a university or research institute as a researcher. The title of his paper was Biological Parameters of Crepe Myrtle Bark Scale, Acanthococcus lagerstromi, on different Lagerstromia species. The authors are Runxi Zi, Bin Wu, Gary Knox, and Meng Meng Gu, and Hongmin Quinn. So,
All right, here we go.
Hello, everyone. My name is Renxi Xie, and I'm a PhD student at Texas A&M University. Today, I would like to briefly talk about my research project on crepe myrtle bark scale. So crepe myrtle bark scale is a non-native pest species here in the United States, and it was originated from East Asia. The top left image here shows you what an infestation of this insect looks like on a small crepe myrtle plant. And the diagram here shows you the life history of crepe myrtle bark scale. So the white sacs here on the plant are actually the male pupils and the gravid females, depending on their size and shape. So in order to investigate the effects of host plants on the life cycle of crepe myrtle bark scale, I conducted several insect rearing experiments using different Lagerstroemia species as the host plants. So basically, I collected eggs from the gravid females here and then incubate these eggs until they hatched into the first instars. And then I will transfer this first newly hatched first instar into the small feeding chambers here, which consist of agar medium and a piece of crepe myrtle cuttings. So these feeding chambers will allow me to document the entire life cycle of the crepe myrtle bark scale. And the life history data will then be analyzed to generate results in terms of the biological parameters, such as the stage-specific survival rate of these insects on different host plants. So as you can see in the graph here, the survival curve of these scale insects are actually quite different on different Lagerstroemia species or cultivars. And therefore, based on this biological and ecological data I collected in this study, better strategies can be developed to control these scale insects in the future. Thank you.
great. Our next speaker is going to be Bin Wu. Bin is a third-year PhD student working with Dr. Meng Meng Gu and Dr. Hongmin Quinn at Texas A&M University. He's also majoring in horticulture. Bin began his education as a student and a research assistant in the Research Institute of Subtropical Forestry, where he was involved with many ornamental plant breeding projects, including camellia hybridization. Bin is now focusing on the interaction of plants and crepe myrtle bark scale, an exotic species mainly found on crepe myrtle. The title of Bin's paper was Green Lacewing, Chrysoperia rufilabris as a voracious predator on crepe myrtle bark scale, Acanthococcus lagerstroemi. The authors are Bin Wu, Runxi Zi, Meng Meng Gu, and Hongmin Quinn.
Howdy. Last year, I happened to find some eggs of green lacewings on twigs of a crepe myrtle bark scale infested plant. The crepe myrtle bark scale was introduced from other countries and now has spread across 14 states of the United States. I was wondering whether crepe myrtle bark scale could be the food source for the green lacewings. So we put a larva green lacewing into a petri dish, which contained lots of fresh eggs, nymphs, and gravid females of crepe myrtle bark scale. Then we found that the larva green lacewing can prey on not only relatively large gravid females, but also some tiny stuff, including eggs and crawlers. Then we did a 24-hour test to evaluate the predator capacity of green lacewings upon crepe myrtle bark scale using around 300 eggs each time. The duration for the first egg consumption varied from 53.2 seconds to 73.2 seconds over the 10 days test. And one green swing can consume about 154 to 196 eggs in 24 hours, which was good. Also, we did a white maize assay under dark using starved larvae green swings over 24 hours to check if they can locate Cretemara barscoe under dark. Interestingly, we found that 78.1% of larvae green swings were able to locate Cretemara barscoe in the white maize assay. So, in this study, we confirmed that Greenland swing is a voracious predator on Cretemara barscoe. For the further research, we might extract and analyze the volatile secreted by Cretemara barscoe, which could attract Greenland swings. Thank you.
Great. Well, that concludes our 4 presentations. So this would be our opportunity to ask any questions, and let's see how we're going to manage this. Do we have anybody that would like to ask a question?
Let me see if I can spotlight all the students. It wouldn't, for some reason, it's not letting me do everyone. But when they speak, they'll pop up in front of everyone.
I'd like to ask a question.
Please go ahead.
On Pingyu, is I'm saying that correctly, Pingyu? On the
Yep.
blueberry research, is that
Yeah.
is that research across all of the blueberry species?
No, it's not. Our goal is to use the sparberry as the blueberry rootstock for like all different blueberry varieties. That's our goal. So that's why we conducted the research to help produce like the sparberry in a large scale. Yeah, that's our goal. Yeah,
Okay.
because I know for breeders, I know in Florida they have people who are doing breeding work to incorporate the traits of sparberry to the blueberry, but they still can't overcome the adaptation challenge. So we were thinking by grafting, maybe we can just overcome the challenge that we're facing right now.
Okay.
Yeah, thank you.
You're welcome to type your question into the chat and someone else will ask it as well if you don't want to be front and center.
Mac, this is Jim Robbins. Can I ask a question of Bin Wu?
Absolutely, Jim.
And before I do that, Bree already beat me to it. It's in the chat, but my just great respect and appreciation to all the students. Just impressive. I appreciate the quality of your presentations, and so thank you for the effort. Bin Wu, a quick question. So I'm really not familiar with the green lace wing. Here in Arkansas, one of our major natural predators is the twice-stabbed lady beetle. Is the green lacewing, is that is it endemic in Texas? I don't know much about it. And do you know how its feeding ability, consumption rate would compare to, say, that twice-stabbed lady beetle? That's my question. Thank you.
Hi, Dr. Robinson. Thank you so much for your question. That's a really good question. Because yes, previously, like Zinan tested the lady beetles on Cretemara barscoe, and I happened to find lacewing feeding on Cretemara barscoe as well. So I did this test. Well, for your question, green lacewing is like a commercial product to control aphids. It's like it sucks and pierce the soft scale. So I think it's kind of similar, but because it's like more, I think it's more voracious because it sucks this like the soft scale very quickly, and yeah, I think it's good. I think when I did this experiment, I also kind of compared lady beetle and green lacewing. I found sometimes green lacewing prey more on Cretemara barscoe, but sometimes lady beetle did better. That's what I found. Hope it worked. Thank you.
So we have a couple of questions in the chat box, and one of them is, what are pine pellets? Aisha, if you wouldn't mind addressing that to our full audience, please.
Yeah, pine pellets are the pellets which are formed from the pine dust, and these are mainly used for like as animal bearings. Yeah, and for like I saw like about the cost differences, and for that we haven't done any cost analysis for that, but we are planning to do that, like comparing the cost for these.
And there's one more question for you that says, did you notice a higher water retention using the paper pellets compared to other treatments?
Yeah, for these pellets, there were as compared to rice hulls, because it's hydrophobic, we didn't find we find differences between that. For pine pellets and paper pellets, yes, they absorb more water and more retention of the water. Maybe, yeah, that may be the situation.
Excellent. Do we have any more questions?
Yeah, I have a question for Ping Yu. You had mentioned growing these sparkberry seedlings and then using those to graft onto blueberries. Is that a proven technique? Do you know like what the grafting percentage is? Like, do they readily graft, or what is the percentage of grafts that take?
So that's a good question. So we, based on our literature review, there are reported articles saying that they successfully use the sparkberry as rootstock for blueberries. So that's because of that, we saw the successful and the potential of sparkberry using as the blueberry rootstock. That's why we did our work to kind of increase the germination rate for sparkberry, because before that, the highest germination for the sparkberry was only 43, which is not desirable for a large scale production for sparkberry. So yes, there are articles about the success, reporting the successful using it as a rootstock, but I don't know the successful. I guess it depends on how you do the grafting process, and depends on the time of year and your rootstock quantity and all that stuff. But yeah, it is successfully reported. Yeah, thank you.
Okay, this brings us to our first presenter. This will be Brooks Parrish. Brooks is a master's student at the University of Florida in the Department of Environmental Horticulture. He's working on ornamental plant breeding projects in the lab of Dr. Zinao Deng at the IFAS Gulf Coast Research and Education Center. He graduated from UF with a Bachelor of Science degree in plant science in 2019. His experiences growing up in production agriculture and conducting undergraduate research have set him on a path to become a plant breeder. And after seeing the impact that new novel cultivars can have on producers, Brooks knew that he wanted to play a part in their development. The title of his paper was Morphological and Cytological Characterization of 6 porterweed Stachytarphetus species selections. The authors on this paper are Brooks Parrish, Renjun Kwan, Sandra B. Wilson, and Zinao Deng.
Okay, can y'all see the screen okay? Okay. All right, hello everyone. Like Dr. Thetford said, I'm Brooks Parrish, and I'll be speaking to you today on morphological and cytological characterization of 6 porterweed Starshita selections. So today I'll be walking you through some information on porterweed and a challenge that is facing the genus. We're going to discuss our plant materials that we use, assess their growth habits in flowering, and look into their DNA content, chromosome counts, and finally, we'll get to the end of our study where we'll look at where the study is going to take us in the future. So when you hear invasive species, it's likely that your mind wanders to things like hydrilla, air potatoes, or even the tropical soda apple. These 3 are among thousands of species that are causing economic, social, and environmental impacts across the globe. According to the US Forest Service, invasive species have contributed to the decline of 42% of US endangered and threatened species. Now, certainly these unwanted plants can cause a lot of problems in our ecosystems, but what about a flowering plant that's sitting in your home landscaping? Some ornamental plants can have these same negative impacts on our environment if given the chance to escape cultivation. Combating ornamental invasion begins with finding alternatives or creating them. Our team of researchers are dedicated to characterizing the invasive potential of ornamental plants and using genetic techniques to identify and create new cultivars that resist invasive species. Now, the project I will be speaking to you about today is on porterweed. Now, if you're in the same boat I am when we started this project, you're probably thinking, what exactly is a porterweed? I quickly learned that porterweed is very much a low maintenance plant. Thankfully, it's drought tolerant and it produces these huge inflorescences filled with these brightly colored flowers. Each inflorescence is capable of producing hundreds of flowers, but only a few of them open at a time in clusters, like you can see in the image on the left of your screen. These plants are easy to find at butterfly gardens where they attract a large range of pollinators like butterflies and hummingbirds. To give some perspective about the genus, there are hundreds of Starchita feta species identified in just Australia and Brazil. These species shown on this slide is Jamaican porterweed. They have these light blue, light purple flowers, and they are our native species to the state of Florida, and they thrive in our southern counties. Unfortunately, Jamaican porterweed does have some serious competition. Nettle leaf porterweed was introduced to the state from Central and South America and has since escaped cultivation. Its rapid increase in abundance has caused it to become listed as a Category 2 invasive species. With all of these flowers, these porterweed are able to spread rapidly, producing lots of seed and have contributed to nettle leaf porterweed becoming invasive in the environment. To make matters worse, researchers have raised concern about the potential for nettle leaf porterweed to naturally hybridize with our native species. This occurrence can have major impacts on the preservation of our native species. So several years ago, back in 2009, Dr. Wilson and her team released a paper titled Seed Production and Viability of 8 Porterweed Selections grown in northern and southern Florida. In addition to the many findings in her paper, the paper identified one cultivar, Naples lilac, to be female sterile. They confirmed that this supposed hybridization between nettle leaf, the invasive, and Jamaican, our native species, is possible. And finally, they identified 2 porterweed selections as diploids, or having 2 copies of their chromosomes. The results of this study led our team to design a further analysis into these species. So the question we set out to answer is, what is there to gain from morphological and cytological data from porterweed? We wanted to know, are there more features that could aid in porterweed identification in the wild? And can we even produce sterile triploid cultivars? And do we even have the materials possible to do so? So we got to work. For this study, we used Jamaican, nettle leaf, coral, and Naples lilac porterweed from the previous study. We added 1 cultivar, dwarf blue porterweed. It was a porterweed that we found at a local nursery near Gainesville, and it was quite popular with a lot of customers. So we wanted to add that to our study as well. And then we also generated that hybrid between nettle leaf, the invasive, and Jamaican, our native porterweed, in the greenhouse through manual pollinations for analysis. And in our study, we denote this cross as U*J3-2. Our plants were maintained in 1-gallon pots for our study with a general use potting media, and we propagated all the accessions through stem cuttings. So before we dive into our internal analysis, we first categorized each of these selections by their growth patterns. The selections fit into 3 different growth habits: upright, semi-upright, and prostrate. Let me see if I can get this to work. There we go. So here on the left, we have coral. We can see that its stems were always moving upright towards the sky. It made this beautiful plant, especially in a potted plant in a greenhouse, of just with the stems going straight up. Nettle leaf had this in between where some of the stems would extend downward, while we also still had that upward growth. So we categorize that as semi-upright. And then Jamaican, our native species, had this prostrate, growing, kind of creeping appearance as it grew across the ground. And then we have our other three. These are our two hybrids and our putative hybrid. So Naples lilac was just like coral, had that upright growth. Dwarf blue had that in between, kind of not as low and sprawling, but still had that low growth as well as upright. And then we had our hybrid between our Jamaican and porterweed, where it took on the growth habit of Jamaican with that sprawling, low to the ground phenotype. So in addition to future breeding selections, understanding these growth habits can give us pretty good clues about the potential species of an unknown porterweed in the wild. As we'll see in a few slides, the colors of all of these flowers are purple, with the exception of coral porterweed. So flower color isn't going to be a huge differentiating factor. So these could be huge distinguishing characteristics. So as I alluded to it, next we're going to talk about flower production. So we evaluated the flower production for each of our different accessions. In the pictures on the left of the slide, we can see we have coral, Naples lilac, nettle leaf, our invasive, our hybrid between Jamaican and nettle leaf, Jamaican, and dwarf blue. Naples lilac and nettle leaf porterweed produced around 65 to 70 flowers per inflorescence. So we consider this flower production to be high in our study, while all the other accessions produce between 10 and 30 flowers. So we categorize these as low production. So we identified two characteristics so far that are important breeding traits and can also be helpful in identification of these selections in the wild: growth habit and flower production. Of particular interest, we want to be able to identify this hybrid between nettle leaf and Jamaican porterweed. We want to identify the hybrid as well as the nettle leaf parent, so that we can identify those in the wild and remove them from our ecosystems. So let's move on to one of the most important plant identifiers: nuclear DNA content. A piece of equipment known as a flow cytometer can be used to estimate the mass of the total DNA in the nucleus of a cell. First, we have to prep our sample. So we add a lysis buffer to a Petri dish and chop a small amount of leaf from our porterweed sample and a small piece of leaf from our internal standard, which in our case was rye. So we use a sharp razor blade, chop up those samples to be able to remove the cell walls and create protoplasts from those cells. Next, we add RNase to our sample, which is going to eat up any RNA that's in the sample, and there's a lot of it. This RNA can cause problems and noise in further analysis. So we have to make sure we get all of that out of our sample. And then we add propidium iodide. This is going to be our fluorescing agent that's going to intercalate with our DNA strands. So when we run it through the machine, this is what's going to fluoresce and be able to give us that estimate of our nuclear DNA content. So then we take our sample and run it through this flow cytometer machine. A laser shines on each of these protoplasts, and the propidium iodide is going to fluoresce that light back, and then we'll be able to, the machine measures that, and we'll get peaks. So on the little computer screen on the right of the slide, you can see we have two different peaks. One peak is going to be for our internal standard, our rye, and then our second peak is going to be for our porterweed sample. We can take the geometric mean of these two peaks and compare them to each other because we know the nuclear DNA content of our rye, our internal standard. So from that, we can compute the nuclear DNA content of our sample. So in this table, we have the DNA content for each of our accessions. While these values alone can't tell us a whole lot about the chromosome number in each selection, we can identify some critical differences in these values. Let's take a look at nettle leaf porterweed. There is a significant increase between this value and UJ 3-2. and an even further jump to Jamaican porterweed. This indicates that these differences are likely due to changes in ploidy level. If we know the chromosome number of a selection, we could use that sample to compare to our unknown and be able to determine the ploidy level. But to determine the chromosome number, we have to use an age-old technique known as chromosome squashing. If we think back on the phases of mitosis and cell division, we may remember that we go through prophase, metaphase, anaphase, telophase, and cytokinesis. During late prophase and early metaphase, the chromosomes become condensed into tightly compact coils that we can stain and make visible using a simple light microscope like the one on our screen. So when beginning this project, we knew that there were previous reports of porterweed having a large number of chromosomes, hundreds of chromosomes. So not only did we need a protocol that's going to give us good staining, but we also needed a protocol that was going to be able to spread all of those chromosomes out so that we could be able to count them without overlaps. So in the past, we used stains such as acetocarmine, which is a red dye, but we had lack of clarity using that with many different species, and it would make identifying this large number of chromosomes very difficult. So instead, we found a protocol used by Chen et al. In 1982, using a GEMSA stain, which is purple. After several rounds of adjustments and modifications, we finally had a refined protocol. So we started with a pretreatment. This pretreatment, we collected a root sample about 1 centimeter long. We placed it in a solution of 8-hydroxyquinoline, which all it does is inhibit the spindle fiber formation that pulls our cells apart during anaphase and mitosis. So this forces all the cells to stop at metaphase when they're most visible for us. Next, we place that in a fixative solution overnight, which basically preserves our cells until we can move on to our next step of our enzyme digestion. So for our enzyme, we cut our root tip even smaller, about 1 millimeter in length, and place that in a 2.5% cellulase and pectinase solution. These two enzymes are critical in removing that thick plant cell wall so that we can view into these cells. After a quick soak in water and a little bit of fixative solution to increase our osmotic pressure, we tore our cells apart on a cold slide, and we're able to tap them apart, get them to spread out, and then we stain those with 2.5% GEMSA. Viewing the cells at 1000 magnification reveal darkly stained and well-spread chromosomes, as you can see in each of these images. So as we can tell, there's not a whole lot of difference between most of these, except for one of our images, dwarf blue down there in the bottom right. Without even knowing the chromosome number, we can easily tell that there is something different going on with this. And knowing that chromosome number of 42, we're able to calculate and know that it has a different base chromosome number as well. This is really interesting because dwarf blue had a higher, it wasn't the lowest DNA content that we calculated in the study, but it has a drastically lower chromosome number. So this is a plot of our DNA content on the x-axis and chromosome number on the y-axis. We can see the positive correlation between We can see the positive correlation between DNA content and chromosome number, which will allow us to use this data to be able to infer the chromosome number of future selections. We can simply know that we have this linear increase in DNA content as we increase chromosome number. So, and then we have one outlier, of course, at the bottom, dwarf blue porterweed. As I said in the previous slide, we don't know what's going on with this specific cultivar, so we're going to need to do some further evaluation to determine its family lineage. So let's take a look at our results all together. We can once again see our selections are separated by growth habit, flower production, that allow our visual differentiating factors. All of our selections have the same chromosome number of 28, with the exception of dwarf blue porterweed that has a base number of 21. And then we can There's our 21. Then we can also verify that our hybrid cross between nettleleaf and Jamaican porterweed, we would expect a pentaploid. So it was very good for us to be able to identify that we do have a pentaploid crossing the 6X with a 4X to be able to get our 5X chromosomes. Next, we're going to look at our nettleleaf porterweed. It has a tetraploid, 4 chromosomes. So to be able to make our interploidy cross, to be able to potentially get a sterile triploid, we need a 4X and a 2X. And we already identified JP's compact, or red compact, and JP's pink in the previous study and diploids prior. So this makes nettleleaf porterweed our primary objective for a triploid hybrid in future studies. So to put this into perspective, our chromosome numbers, nuclear DNA content. DNA content can be determined in as little as 10 minutes. We can run those right through the machine. Chromosome counting is a 2-day procedure and very laborious. So having these values to be able to use for future evaluation of other species is going to be critical. So let's take a look back at our question. What is there to gain from morphological and cytological data from porterweed? Based on the results of our study, there is certainly much to gain from these analyses. Differences in growth habits, flowering, and nuclear DNA content can all be used to differentiate porterweed accessions and provide better means for identification of unknown species. One of the most popular ways of creating sterile plants is through the production of triploids between a tetraploid and a diploid cross. We saw earlier, JP's pink, red compact are our diploids, and then we identified nettleleaf as a tetraploid, and that's going to be our primary candidate for this cross. And then in addition to our flow cytometry, our chromosome squashing protocol will have major impacts on other ornamental plant breeding programs. Flow cytometry equipment costs thousands of dollars. So low-income ornamental plant breeding programs don't have the luxury of buying this expensive equipment. For chromosome squashing, all you need are a few reagents and a simple light microscope, and you can be able to get this valuable information. So it's in our hope that our protocol will be adapted to other species and other programs. In our lab alone, we've already adapted it to Caladium, Lantana, and many other species, and still get that beautiful spread of chromosomes. Now that we have all this important information, all that's left is for breeders to go out, use these valuable information that we've given them, and go out and make these triploid crosses. I'm looking forward to the near future where sterile porterweed cultivars are filling our landscaping and the threat of invasion is long gone. Thank you for your time today, and I look forward to any questions you have.
Very good. Thank you, Brooks. So did we have any questions from our audience today?
I got one. Can you hear me, Matt?
Yes, sir.
Yes, sir.
I want to know, in your final summary, you had chromosome number, and then you had a base chromosome number. Now,
yes, sir.
I'm an old school guy here. What are we talking about? Base and what are we talking? Chromosome's a chromosome, isn't it?
Yes, sir. Absolutely. So our base chromosome number is our one group of chromosomes in our cell when they're dividing. So we talked about having a tetraploid. So our tetraploid means that we have our 4 different copies of chromosomes. So one copy of all those chromosomes would be our base chromosome number. And then we also have 3 additional copies. So our traditional diploid, just like us, we have 2 copies of our chromosomes. So our base chromosome number is half of our total chromosome number. I hope that answered.
I want to go a little bit deeper there. In this one, it was 168 chromosomes, but the base chromosome was only 24. Now, how do you do that kind of math to get that kind of number?
So we have to use our information that we have from previous of other species. So we know that other species with lower numbers of chromosomes, like we identified our tetraploid, and there's other information on some of our diploids already. We have to go to our lowest possible chromosome number of being able to get to that base chromosome number. We knew that that one had 6. The next lowest denominator would have, or the lower base chromosome number would have identified that as a pentaploid. And we would have really gotten to some bigger numbers. So that we had to settle with that as our base chromosome number.
Thank you very much.
Yes, sir. Thank you.
How does porterweed spread? Is it spread through seeds, or is it like a vegetative plant that spreads through runners?
Seed primarily. Each of these flowers will produce tons of seed, especially the nettle leaf, that invasive variety. Even seeing some of it in landscaping in homes near where I live, you'll walk by and sure, you'll see beautiful flowers, and then you'll go back a few weeks later and you just see seed pods popping everywhere. So it spreads very rapidly through seed.
Okay, thanks. I have one more quick question too. So you recommended trying to use these techniques, the chromosome squashing for, small mom and pop breeders, we could call them. What advice would you have for those breeders about this process? I've never heard of it before and it sounds interesting, but what are some of the ups and downs of this process?
Chromosome squashing can definitely be a technique to master. Adjusting the different times. Mainly, we have the pre-treatment time in our 8-hydroxyquinoline to inhibit our spindle formation. That time will vary based on whatever species you're working with. Mainly, we've found due to root size. So if you have larger roots, like I work with a lot of caladiums, and those require much longer, 5 or 6 hours of pre-treatment time, as opposed to porterweed took around 4, lantana will even go down to 3 hours. So really playing with a lot of different combinations of each of these different steps and doing trial and error until you find it, because once you find you're perfect for your species, it's smooth sailing from there and you get beautiful spreads every time. But for mom and pop breeders, low income ornamental breeders, this information is critical, especially if you spend a lot of time making crosses, especially interspecific crosses. A lot of the more difficult where embryo rescue and things like that might be involved. Knowing that chromosome number and if it's even feasible for the plants to be able to be compatible is very important.
Yeah, I agree. It's crucial. Nice presentation.
Thank you.
Mac, I set a timer for 5 minutes, so he would just have 5 minutes of questions unless we have, yep, just 5 minutes. So there's 30 seconds left. We got room for one more.
All right. Anybody else?
Brooks, this is Jim Robbins, University of Arkansas. Great presentation. Hey, I got a quick question. I was intrigued with the Naples lilac hybrid compared to the UJ3-2. The Naples lilac showed what I would expect a hybrid. It had the flower size. Just interesting that the flower size on your other hybrid was it was smaller and the flower production was rated as low.
Yes, sir. Naples lilac has proved to be very, very prolific growth habits. It produces bigger plants, bigger flowers. So it's really production or the product of a great cross. And it's our top candidate for replacement with our nettle leaf invasive porterweed. Our cross between nettle leaf and Jamaican, yeah, I completely understand where you're coming from. We did not see expected, hybrid vigor like we did see from our Naples lilac porterweed, which I would really be interested to find the female parent of the Naples lilac and be able to do some studies with that as well to see what kind of traits it's bringing from that side as well.
Okay. Thank you, Brooks. We appreciate your time.
Thank you.
All right, then I will introduce our next speaker. Our next speaker is Yuvraj Kamare. He is a PhD student at the Department of Environmental Horticulture at the University of Florida. He completed his Master of Science at the University of Teesside in the UK. His current area of study is focused on weed management and ornamental and landscape plants. And his research focuses on developing new methods of weed control by strategically improving the cultural practices used by nursery growers. The goal of his research is to find effective, economical, and environmentally friendly weed management options. After graduation, he plans to work as a successful weed scientist with an agrochemical company or in academia, helping to find and develop new methods of weed management. The title of his paper was Influence of Substrate Stratification and Fertilizer Placement on Growth of Ligustrum, Ligustrum japonicum, and Germination and Biomass of Bittercress, Cardamine flexuosa, in containers. The authors on his paper were Yuvraj Kamare, Christopher Marvel, James Altland, and Annette Chandler.
Oh, sorry about that. Can everyone hear me now?
Yes.
Okay. Hello, everyone. Thank you for that introduction. Without taking any more time, I'll just jump into my presentation. So as most of us know, weed management is one of the costly and challenging aspects of container nursery. On an average, a nursery grower spends about $11,000 per acre per year just to manage weeds. And research has shown that even a single weed can reduce the crop growth by almost 40 to 60% in just one season. So to fight this, nursery growers depend heavily on herbicides and supplemental hand weeding. While herbicides are effective, the options are really limited as there's no post-emergence herbicide available for container-grown crops. And there's only limited amount of pre-emergence herbicides available. And if it comes for a small nursery, it's really costly to pay for those herbicides. And it's really difficult as most of the herbicides are granular and applying to a container is difficult. Especially in Florida and places with sandy soil, they have runoff issues and other environmental concerns like leaching into irrigation pond or groundwater. Like I said, along with herbicides, the growers also rely on supplemental hand weeding. But on an average, a nursery grower spends about $4,000 per acre per year just to manage weed through hand weeding. And in today's world, hand weeding itself is expensive. It's a problem itself to find labor, it's laborers and time consuming. So what could be a potential solution for this? Due to the cost of hand weeding and challenges associated with herbicides, a more integrated weed management strategies are needed for nursery production. So one of the strategies can be stratifying substrate, or you can simply call it layering to create designer substrates. So the objective of stratifying substrates is to determine ideal substrate characteristics needed to reduce weed growth and its germination, while at the same time not affecting the ornamental plant growth and its quality. So here's a diagram that would give us a better idea of how stratified substrates work. So at the top layer, the larger particle materials are added, which has no sand or peat or fertilizer to it, while the bottom layer consists of the standard size substrate. So with this, as a result, the top portion of the substrate does not hold enough moisture. So the weed seeds that fall in, they do not germinate as there's no moisture available over there. That's why the objective of this study was to evaluate the effect of stratified substrates and strategic fertilizer placement on the growth of common nursery weeds and ornamental crops. Substrate stratification has shown promise to prevent weed growth. It is a good non-chemical option. It has shown that it increases the moisture retention in the substrate profile and also improves the plant growth. So this entire study was conducted at Mid-Florida Research and Education Center in Apopka, Florida. The weed species we chose was bittercress, and the ornamental species was ligustrum. The substrate over here consisted of pine bark and control is fertilizer. So we used pine bark of 3 different particle sizes. The pine bark was as small as 0.95 centimeter, followed by 1.3 centimeter, and the largest one was 1.9 centimeter. And also we added a fourth particle size again with 1.9 centimeter, but without any fines in them. So this way we had 4 different particle sizes. So this is an illustration of how all the treatments looked. So the TO stands for throughout, and S stands for stratified substrates. So I'll explain it much better. So we had 0.95 centimeter, 1.3 centimeter, and 1.9 centimeter of substrate throughout the profile, throughout the container. And it had a fertilizer incorporated throughout the substrate profile. So this way we had 3 incorporated treatments. And the remaining consisted of stratified treatments that were either 1.3 centimeter or 1.9 centimeter particle size pine bark. So the top layer was 1.3 or 1.9 centimeter pine bark, and the bottom layer consisted of the industry standard 0.95 centimeter pine bark. And the top substrate of 1.3 and 1.9 was applied at a depth of either 5 or 7.5 centimeter. So this way the top layer had bigger particle size and had no fertilizer. So only in the stratified substrate, only the bottom layer had fertilizer. The top layer had no fertilizer, no peat, no sand. So here's a closer look of 2 treatments side to side. So this is a standard 0.95 throughout substrate. So TO stands for throughout. So as you can see, the fertilizer is incorporated throughout the substrate profile, and there's fertilizer even at the top surface of the container. And the second one is stratified, again, 1.9 centimeter, S stands for stratified at 7.5 centimeter depth. So you can see the fertilizer is at about 7.5 centimeter depth over here. So the top profile has no nutrients for weed seedlings to access. So for this study, we seeded all the plots with 25 seeds of bittercress. And for bittercress, we did the germination count at 4 and 10 weeks after potting them. And at the trial conclusion, which was at 10 weeks after potting, we did the shoot dry rate of bittercress. And for our ligustrum, we measured the growth index every 2 months. And at trial conclusion, which was at 6 months after potting them, we measured the shoot and root dry rate. So moving on to our results, here's a picture of bittercress after we potted them, after 2 weeks after potting them. As you can see, these 3 treatments, 0.95, 1.3, and 1.9, which are throughout this, which is an incorporated treatment, had good germination of bittercress, while at the same time, there's few to none in any of the stratified treatments. So this is a graph that follows the picture I just showed. So this is germination of bittercress after 4 weeks after potting them. So x-axis over here stands for all the substrate treatments. So 0.95 to TO will be 0.95 centimeter throughout the substrate. And 1.3 S5 will be 1.3 centimeter stratified at 5 centimeter depth, and so on. So over here, we measured the bittercress count per pot. So bittercress germination was seen highest in the 0.95 incorporated treatment and 1.9 incorporated treatment, while the germination was lowest in all of the stratified substrates. We saw that the lowest germination was observed in the 1.9 stratified substrate, which had no fines in them. Here's a picture of bittercress, the same bittercress after 10 weeks after potting them. As you can see, there's substantial growth of bittercress in all the three incorporated treatments, 0.95, 1.3, and 1.9. And there's not few to none in any of the stratified treatments. Again, here's a graph of the shoot dry rate we took after 10 weeks. So all the stratified treatments had significantly lower dry rate compared to all the incorporated treatments. So moving on to our cash crop, that's ligustrum. So here we measured the growth index at trial conclusion, which was at 6 months after potting them. So the growth of ligustrum in all the stratified treatments, it was similar to the incorporated treatment. There were few variation in growth with the lowest in the stratified of 1.9 centimeter pine bark, and the highest was seen in 1.9 centimeter pine bark without any fines. And here's a graph of ligustrum shoot dry weight, which was measured at six months after potting them. Again, it's the same treatments at our x-axis. So here, all the stratified substrate had similar shoot biomass compared to the 0.95 incorporated treatment, which is this first one. But there was a difference at 1.9 stratified substrate at 5 and 7.5 centimeter, which had a 30% decrease in the shoot dry weight if you compare it to the 1.3 incorporated treatment. So here's a picture of all the ligustrum plants at trial conclusion. And as you can see, all the plants reached a marketable size in almost all of the treatments. Again, we did the root dry weight, and this is a picture of dry roots in all of the treatments. They look healthy and good size in all of the treatments. So overall with Bitter-Cress, we saw that the germination was highest in the industry standard of 0.95 incorporated treatment, and it followed by the 1.9 incorporated treatment. And we noticed that the germination was lowest in all of the stratified substrate. We saw almost only 15 to 30% of wheat germination of Bitter-Cress germinated. So at trial conclusion, stratification and strategic fertilizer placement, it resulted in a decreases ranging from 80 to 97% in Bitter-Cress biomass. And overall for ligustrum at six months after potting them, all the stratified substrate had growth index similar to the standard incorporated treatments. And all the stratified substrate also had similar root and shoot biomass when you compare it to the industry standard 0.95 treatment. So as I said before, there was some difference. There was at 1.9 centimeters stratified at 5 and 7.5 centimeter depth. So it had a lower shoot and root weight compared to the 1.3 incorporated treatment. It had some 25 to 30% lower growth. But surprisingly, 1.9 stratified treatment, which had any fines in them, without any fines, it had the highest shoot and root dry weight. It was similar or almost greater than all of the incorporated treatments. So in conclusion, we didn't see no difference were observed between any of the stratified substrate and an industry standard substrate by the time plants reach marketable size. And the growth of Bitter-Cress substantially decreased in the stratified substrate. This indicates that stratified substrate could potentially be used as a part of an overall weed management strategy. And this study can be utilized as a weed management tool in crops, especially crops that are intolerant of herbicides. So for our future work, we'll be determining the effect of these strategies on the growth of other common ornamental species and also other problematic weeds in container nurseries. Also, we'll do the same. We have the plan to do the study the effect of these methods on irrigation and fertilizer efficiency. And this will be a good strategy to combine with other non-chemical techniques. So we have an ongoing study right now with liverwort. And as you can see over here, liverwort has grown very good in all the 3 treatments of incorporated treatments, 0.95, 1.3, and 1.9. But there was no growth in any of the stratified treatments. That's all from my side. Thank you.
Thank you. And at this point, we'll see if we have any questions.
I got one. This is Charlie Parkinson.
All right.
Can I go?
Yes, sir.
I want to find out, how did you plant the ligustrum in this substrates? Did you have it, tell me how you did it, how you planted the plants?
So we, while planting the ligustrum, we had all the base layer first. We had the bottom layer first over there, and then we just placed the root ball and added the top layer on top of it. So this way, at 5 centimeter, the ligustrum was touching the bottom layer. The root ball of ligustrum was touching the bottom layer of the containers.
But it wasn't incorporated in the bottom layer.
No, because
Okay.
the root ball were only 5 centimeter in size, so they would just get to the base
Okay.
of.
Thank you.
Okay, thank you.
Jim Robbins, I've got a quick question. Can you think this through in a production setting, how on a potting line this would work? Would you have to modify existing, particularly like some of the auger? Have you thought this through, again, how this might be incorporated into our, would we have to adapt existing methods, or so what are your thoughts?
I think there are machines. I mean, usually there's most of the time there are two people potting in there. So one machine fills in the soil, and then one guy puts in the plant and then fills up the pot. So if to incorporate this, we'll need an additional worker in there, or even two pallets of the machine, which fills in bottom layer, and the other one fills in the top layer of the container. Does that make sense?
Yes, thank you. And great presentation.
Thank you.
So we have a question in our chat, and the question is, did you happen to take any pour-through measurements with the different treatments? Do you have any idea of what's going on there?
We are planning to do it. We haven't done it yet, but we are planning to do the pour-through measurements of all the substrate.
Anybody else have any questions?
Yeah, I have a quick question. Do you know how your findings, or how using a larger substrate at the top of the container, the stratified strategy, I guess we could call it, do you know how that compares to rice hulls? Have you guys looked into using rice hulls as a top dress?
We haven't used right now we're just using pine bark, because you can get different sizes in pine bark, but with rice hulls we haven't used any. We haven't tried the rice hulls yet. But I would think it'll be difficult to get different sizes of rice hull, just like pine bark, you can get different sizes at 1.9, even at 2 centimeter.
Right, yeah, I was just mostly curious if you had seen other studies that had looked at if rice hulls prevent weed seed germination, and if the rates were, comparable to a stratified bark media.
There's rice hulls have been used as a mulch, but this hasn't been used as a stratified yet. So there's no study done with stratified substrate using rice hulls yet. So it'll be a good idea to try it in next project on it.
Who else? Come on.
I know we have some other students out there participating today. Maybe we can throw out a challenge for a question from one of them, please.
Some of your students, perhaps, Mac?
I know there are a couple of mine out there.
Yuri, this is Jim Robbins again at University of Arkansas. Hey, how did you water these with a disparity in particle sizes? Is it hand watered overhead? Is that the method?
No, it was a sprinkler irrigation, 1.3 centimeter twice a day. So we mimicked the nursery condition in there. So instead of hand watering them, we just kept it overhead sprinklers.
Okay, so they were all given the same application amount. That was not varied. Okay, great. Thank you.
I actually have a question. I know we're out of time, but I'm gonna ask it real quick.
You're in control.
On the ligustrum, did you weed those pots as they were growing?
Yes.
Okay.
There wasn't much need to weed them, especially they're mostly with the incorporated treatment. I had to go and weed them, but with the stratified ones, I noticed that there was not much weeds going on in there.
Okay. Oops, sorry, that's me. That's one of our speakers for the summit. Who has a question? I'll call him back.
Okay.
Okay, do you want to take a 5-minute break or do you want to?
We have one more speaker. Why don't we do
Okay.
that one and then we can look at the break.
Okay.
All right, that brings us to our next speaker, and Brian Schulker. Brian is a PhD student at North Carolina State University. He completed his bachelor's degree at the University of South Florida, majoring in environmental science and policy, with a 2-year position at the USD Agriculture Research Station, Horticulture Research Laboratory, following his graduation. Both his MS and now his PhD share a focus on substrate water and nutrient movement, capture and retention, with the overall goal of understanding how substrate components will affect water dynamics within a container. After graduation, he hopes to find a position as a research professor at a university or in industry to continue working on the advancement of soilless media. The title of his paper was "Where Did the Water Go?" Authors on the paper are Brian Schulker, Brian Jackson, and William Fontenot. Brian, please.
Thank you. Let's make sure I'm sharing the right screen. Is this the right screen? Okay. Hello, everyone. As he said, the question that we're going to hope to answer today is, where did the water go? So before we move completely into this research, I want to give you a little background on the current state of water, substrates, and irrigation techniques. So starting with water, around 70% of the world's water supply is used for agriculture, and that's a statistic given to us by the FAO. Sustainability of our global water supply is critical as global population continues to increase, with water shortages already starting around the world. Putting this in a bigger perspective, population increase and global infrastructure are at constant odds with water resources. Poor water management at the greenhouse and nursery level will equate to a lower water use efficiency. And now a study done by Beretti in 2019 resulted in global water demand for all uses is predicted to increase by 30% by 2050, while global water demand for agricultural water use will increase by 60% in 2025, and that's with the same study. To put this in a smaller, bigger perspective, a few years ago, the Clean Water R3 grant was put into place. This is an $8.7 million NEPA grant, and at the time of me looking at this, there was 30-ish publications, and there could be more now, and there was 11 partnering universities. As someone who focuses almost all of their research on substrates, I would be remiss not to talk a little bit about them. So starting off, I want to give you a little overview on just common substrates in the industry. Now, after we just went through those other presentations, you've seen some other ones. But so we've got sphagnum peat. It's technically the most used component in horticulture, and it's known for its high water holding capacity. Now, coconut coir is manufactured from the coconut husk. It's known for its high water holding capacity and re-wettability, and also for its occasional high salinity, as coconut trees are usually in the tropics, and there's salt water down there as well. Pine bark is a major nursery substrate. It's known for its high particle size diversity. It's sustainable, and as you saw in the last presentation, it's versatile, where there are different manufacturing processes for different particle sizes, and it's used often. Contrary to those three substrates, we have additives like perlite, which is an aluminosilicate volcanic rock. It's used to improve aeration and drainage, Now there is no known water holding ability, but it's also lightweight to use to add to those substrates. Similar to perlite, we have wood fiber. There are multiple refinement techniques you can do for wood fiber. There are some water holding abilities because it is a porous material. It's cheaper than perlite, it's sustainable and renewable as trees do grow back. Looking now at this table you see in front of you, there are a lot of statistics on there, but we'll work through them. This table illustrates a survey of soilless media usage in 2017 presented by Chris Block at the International Peatland Society Conference. And there's a projection of use by 2050 of these materials and the volume that these materials are expected to increase. So the volume of both wood fiber and bark is predicted to increase by over 1,000%. Peat in 2050, as you can see, there's 2 question marks next to the 80. This is because there's a raising environmental concern of peat harvesting in Europe and the potential political changes that could affect its use in the future. Now, available arable land is slowly dwindling, and increases in land area could require deforestation. Increases in substrate advancements are necessary to sustain this production and increase production with lessened land area. Now, the research I'm going to present to you is focusing on sub-irrigation, and yes, there are more irrigation tactics, but to put some of them in perspective, we have flood floors, capillary mats, and ebb and flow systems. Ebb and flow systems can be smaller or larger. The picture you see there is the entire floor. There's also flood floor systems, which are using a similar tactic of raising the water. And then we have capillary mats. We're using the weight of the pot to kind of create a dimple in that surface there to allow the substrate to imbibe water through the pot. Now, these practices usually just allow water to flow up, settle for a while, allow the substrate to imbibe water, and then redrain. Pulsing is a different tactic to this altogether. Surface irrigation methods, for reference, are also known to have increased runoff. So if you're using sprinklers or emitters, water moving through, if it's not captured, it moves out. One of the ways to reduce this water loss and runoff is to pulse apply your water. What I mean by pulse apply is by using shorter bursts of irrigation instead of one long application, so that the substrate can slowly become preconditioned to capture this water. This has been researched in surface irrigation methods, but not little has been shown in sub-irrigation. The objectives of my research are to understand how pulsing through sub-irrigation affects the initial capture rate using 3 substrate components under 3 moisture conditions with 3 irrigation water depths under 3 pulsing times. The 3 substrates we used for this project were coconut coir, pine bark, and peat. We used the 3 moisture contents, as I said before. 50% was used as a recommended moisture at potting. That's usually what you get from the bag. 33% is a simulated drier moisture condition if you allow your substrates to dry down, while 67% is a simulated higher moisture condition. So we can test all 3 of these. The pictures that you see now in front of you this is the ebb and flood sub-irrigation system that we used for this experiment, and it was designed and engineered here at NC State in the Hort Substrates Lab. On the left, you see the fully deconstructed 3D model of the system, while on the right, you see the construction process, including the metal rings and the weight, the mesh screen shown in black, the system, and then the fully constructed system itself. We use these rings, layering the bottom below the mesh screen to elevate that screen off the surface of the system and optimize the surface area exposure to water. And now you see what they look like in person. On the left, you do see it completely deconstructed, as I said before. And on the right, you see the mesh screen on top of the aluminum rings with the weight placed in the middle, because this is a plastic system itself. So we're trying to make sure the entire bottom is flat. So to accurately identify how irrigation exposure affects substrate water capture, we use 3 pulsing times of 5 minutes, 10 minutes, and 20 minutes with irrigation events that add up to a total of 60 minutes. So if it's a 5-minute event, we're doing 12 of them to add up to a total of 60 minutes in time. And at 3 irrigation water levels of 2 millimeters, 20 millimeters, and 35 millimeters, or in inches, it's 0.08, 0.78, and 1.38 inches. 2 millimeters we used to simulate capillary water capture of the substrate only, so we're not flooding it. Water is held at the very base of it and allowing the substrate to do all the work for us. 20 millimeters represented a fifth of the cylinder itself, and then 35 represented a third. And the 35 millimeters was generally found to be used as an irrigation practice. After all these substrates are packed, we pack them to 10 centimeters and are placed on the mesh screen, as you see in that bottom right photo. Once placed, the unit is then filled with water. We filled up to whatever method we were doing at the time, either 2, 20, or 35 millimeters. Water is filled up to a level controlling copper pipe. Once water spilled into the copper pipe, that meant inflow equaled outflow, and so we could start the timer then. This timer was not started beforehand because we wanted to make sure that all things were equal. Once the 5, 10, or 20 minutes was up, the system is drained, the cylinders are weighed, and then the process is repeated for however many events were necessary. Statistics were also done on this experiment using Tukey's honestly significant difference test with an alpha of 0.05 in SAS. Moving into the results, initial capture rate, container capacity, and particle size distribution were done on these samples. For brevity and 20-minute time constraints, I'll only be discussing the initial capture rate and container capacity. So initial capture rate here was defined by taking the capture rate, the initial capture, subtracting it from the previous capture, and dividing it by time. For this experiment, because we were only covering the initial, so the first capture, we used the initial volumetric water content, so the volumetric water content of the moisture content. So if it's 33, 50, and 67, those have different initial volumetric water contents as the CP value in that equation. And again, all of this is divided by time. So now looking at the results, we're starting off. This figure represents the initial capture rate of coconut coir at all water depths and moisture contents. The y-axis represents the initial capture rate in milliliters per minute, with the x-axis being split into 3 sections for 33, 50, and 67% moisture content at water levels of 2, 20, and 35 millimeters. The blue bars represent 5-minute pulsing. Orange bars represent 10 minutes, while gray bars represented 20 minutes. I want to start here at 50% moisture, as this was the middle ground. It's evident that coconut coir did not have any trouble capturing water, regardless of water level, with the initial capture rate increasing as water depth increases as well. Decreasing the moisture content down to 33% does not result in a major change in the initial capture rate of coir. The same sentiments do hold true for capture rate increasing as depth increasing, and with decreasing moisture content affecting it about 1 to 2 milliliters per minute lower than 50% moisture content. Now finally increasing to 67% moisture, this decreased the initial capture rate slightly compared to 50%. However, the take-home message here for coconut coir is that it was not greatly affected by pulsing time or moisture content and can capture water under drier conditions. Now, putting this entire figure here in perspective, it shows you based on the bars that as pulsing time increases, water capture decreases. But think of that number as an average capture rate, again, in milliliters per minute. Remember that 5 minutes and 10 minutes and 20 minutes, that's the divisor in that equation. So if you look at it in that regard, it'll show you that pulsing had very little effect on coconut coir, capturing more water as depth increased, but total water was very similar across the board. Moving to peat shows you just how impactful moisture content weighs on the ability of peat to capture water. Peat is known to be hydrophobic under drier conditions, and that's evident even at 50% moisture, with capture rate values increasing as depth increases. But if you compare it to coir, it's still about 8 milliliters per minute lower under equal conditions. Hydrophobicity proved to decrease water capture severely at 33%, barely breaking a 0.5 milliliter per minute, even at 35 milliliters of water depth. This just shows how important it is to prevent peat from drying down in a growing operation, as rewetting can prove to be difficult or even impossible. Now, increasing the moisture content to 67% allows peat to capture nearly 30% more water than under lower conditions. While this is still, again, 4 milliliters per minute less than what coconut coir did in the same conditions, this exemplifies peat's true water capture abilities. Lastly, we looked at pine bark. It showed that nearly no treatment effects across all water depths. Pine bark does have the highest percentage of large particles, increasing the pore sizes within the substrate, which may be a leading factor in its initial capture rates. Reducing moisture content to 33 impacts this initial capture rate. However, at 2 millimeters, so the lowest water level, it was able to capture the most water compared to 20 and 35 millimeters, which is contrary to both peat and coir. And then increasing to 67%, pine bark still showed very little difference based on pulsing time and water level. While the initial capture rate values are higher for 67%, it didn't fluctuate in this capture rate based on treatment effects, as did both peat and coir. So now we're gonna look at the container capacity. This figure is a histogram representing the container capacity for each material at 3 tested moisture contents of 33, 50, and 67. Looking at the y-axis, it's the container capacity in percent volumetric water content. The rest of the figures we've been looking at were in milliliters per minute. On the x-axis is peat, coir, and pine bark from left to right. Blue represents 33% moisture, orange will represent 50% moisture, and gray will represent 67% moisture. Starting at 33, similar to our results for capture rate, peat by far is the lowest container capacity, and that follows our results, like as I said before, with both coir and pine bark having higher container capacities under low moisture conditions. At 50%, the container capacity of peat increases by 30%, with coir and pine bark, again, showing very little change. And then at 67, it's clear that both coir and pine bark were unaffected by treatment differences, while peat was affected by moisture content. And then notably for this, the pine bark we used in the study was specifically engineered to have a container capacity of 55%. And while it is obviously variable in nature as bark particles can be non-uniform, this seems to be very close to right on target. So based on these experimental results, initial moisture content prior to the first irrigation event and depth had the greatest effect on the initial capture rate of peat, coir, and bark. Across all moisture contents and water depths, coir was able to capture and retain the most water comparatively. However, moisture content played a role in determining just how fast it was able to do so. As water level increases, so does the capture rate, showing that an increase in irrigation water depth truly plays the biggest role in the amount of water coir captures in that first irrigation event, increasing by 5 milliliters per minute in between the lowest water level of 2 millimeters and the highest of 35. And then initial moisture content has the greatest impact on the ability of peat to capture water. Regardless of pulsing time or water depth, moisture content had the largest impact. As you increase those moisture levels in peat, the substrate's ability to capture water increases, but peat is still known to be hydrophobic, and it truly hindered the ability of peat to re-wet, taking more water and time to wet that substrate. The main result here was showing that the initial capture rate is nearly 10 milliliters per minute less under low moisture conditions, representing the largest difference of all 3 substrates. And then pine bark, as I said before, was comparably unaffected by pulse time and observed an increase in initial capture rate as water depth increased. Similar to peat and coir, the higher the moisture content and water depth, the greater the capture. And some conclusions here. Initial capture rate here was designed to be able to understand that first irrigation characteristic of these substrates and how different variables such as water depth and pulse time can affect the amount of water captured by these substrates. The results showed that initial moisture content played the biggest role in the ability of these materials to take up water. Peat showing the greatest difference again, coconut coir capturing and retaining water in almost like a sponge, like fashion, and then pine bark showing very little variability based on that moisture content. But overall, these three substrates represented different abilities in their water capture through initial moisture content, water level, and pulse time. So wrapping everything up here, the preconditioning of peat, core, and pine bark, so the initial moisture contents of 33, 50, and 67, made the biggest impact on these initial capture rates. Keeping the substrate from drying down can overall improve substrate water capture and then reduce the amount of water needed to irrigate. And then in the future, engineering substrates with the goal of water capture efficiency could increase the precision of future plant production. And with that, thank you.
Thank you. Do we have any questions for Brian?
Can I have a quick question?
Sure.
Absolutely.
Yeah, thank you so much, Frank. That was a very nice presentation. I just wondering, how would you incorporate your research into real container plant production, since we know, like as you said, your peat, the container capacity of the peat was lower than 50%, which is not ideal for container substrates, because based on Dr. Yeager's research, the ideal or suitable range for container substrates, container capacity was 45 to 65. So the lower, so for your peat, it was lower than 50. So how can you incorporate your research data or results into the real container plant production?
That's a good question. And the overall idea of this project was to do all of these substrates without any additives. So peat in the real world is usually going to have a wetting agent involved, or it's going to be a mix with perlite or wood fiber. That'll increase the container capacity of these materials. Peat has a variability in its container capacity, like you said, between 45 and 60, and I've seen it as high as 70. So the materials that we used here showed these showed characteristics like this, but that doesn't mean that elsewhere the materials could be different.
Thank you.
How did you engineer your pine bark substrate to have a particular moisture content?
I wasn't the lucky enough one to do that for me. We got our pine bark from Pacific Organics here in North Carolina, and we had materials of Airspace 40 or CC 55, and for this project, I wanted to see how accurate it could be with a container capacity of 55, and I think the results showed that they're right there.
Thank you.
Brian, Jim Robbins, University of Arkansas. What's the particle size range on your pine bark?
The pine bark, we had I did a coarse, medium, and fine particle sizes. So the particle sizes ranged from 2 millimeters all the way down to 0.11. 60% of those particles were between 2 millimeters and 0.5 millimeters.
Charlie Parkinson here. I got a question. On one of your first slides, it showed soils and /tufts, TUFFS. What is tufts?
I don't know where I have that.
Well, it was where you said the pine bark had increased by so much, and you had.
Oh, yes. The chart that I put up there. Tufts, I don't have that answer for you, unfortunately. This was just something that I brought up to show the top 6 substrates, and I tried to ignore outside of the substrate realm.
Thank you.
Anybody else with a question?
Jinx, you owe me a Coke.
So Brian, I have one for you. there seems to be a lot of interest lately in the use of some of the whole tree products, the really fine material that you mentioned earlier in your introduction. How do you think that product would compare amongst the three products that you actually tested?
So whole tree has its own substrate?
Yeah, when it's ground to a really fine texture as a peat substitute, how do you think it might compare to what you tested?
That's a good question. It really depends on how you grind that down, because it'll probably be in a very similar category as if I tested wood fiber under different conditions and use that as its own substrate. Now, we have done wood fiber as its own substrate and tested the conditions there. Though it's such a light material and it doesn't always hold water nearly as well as peat did or core. So I would have to get back to you on that answer. I don't want to give you something I don't remember.
Good. Last opportunity for some questions for Brian. Well, thank you very much. Well, that concludes our three students in the competition section here. And so I want to thank everybody for their great presentations. And I see there's some applause coming up on the screen. You all did a great job. And I think we did a good job of doing this in a virtual setting.
Greetings from Southern Region IPPS in North Carolina. This is Kevin Gann at Hefner's Nursery. Southern Region IPPS Education Endowment was established in 2017 to support the educational activities of the Southern Region. Currently, the endowment can support the following: the Charlie Parkerson IPPS Student Research Awards Program, the Vivian Mundy Young Horticultural Professional Scholarship Work Program, the Early Career Propagator Exchange Program, subsidizing student attendance at our annual meetings, and subsidizing our educational needs of the Southern Region. More information about these programs can be found on our website. We have partnered with the Horticulture Research Institute for our endowment. HRI is a 501c3 tax-exempt organization, and contributions to the endowment are tax deductible. Our endowment is a temporary restricted fund, which allows us to spend monies from the corpus. The endowment is also able to accept stock donations. Funded initially by a $20,000 anonymous donation in June of 2017, the endowment has grown to over $71,000 in 3 short years. Our current goal is to grow the fund to $100,000 by 2023. Please consider contributing directly to HRI with the link provided to help grow our endowment and perpetuate the Southern Region IPPS education efforts.
Well, I just want to mention that it's really exciting to hear how much the endowment has grown. And as you can hear, there's a lot of programs going on with IPPS in the Southern Region that are supported by this. And today we're going to hear a little bit about some of those programs. And I'm going to turn it over to Judson LeCompte to introduce to us some videos from some people who have been participating in these programs. Judson.
Thanks, Mac. I really want our students to focus in on a lot of these great opportunities. I have been very fortunate to have received every one of these. So first, we're going to talk about the Vivian Mundy Scholars. This is an opportunity for students to come to our conference. It gives you a travel stipend and it makes you work the conference, but it's such a good opportunity. It really forces you to have that uncomfortable situation and meet growers and meet researchers. Is definitely an opportunity that you all need to take part in. And then our exchange program with the European Union. I was the first exchange recipient and I got to go to England and see all these great nurseries. You'll hear about it in just a second. But Bree was also a recipient, and Leanne. We also look for hosts. So any of our growers who would like to host a European member, it is a great opportunity for you. Students, I paid for nothing, zero, absolutely nothing. So make sure you apply for that. And then finally, what you saw today, which we normally do in person, the Charlie Parkerson Student Research Competition. Again, it makes you experience a little uncomfort and get a little nervous and get a little anxious, as I'm sure most of our students can relate to, but it is great training for you in your future. And for our students, if you want to find out more about that, go to our website at srna.ipps.org. Is that correct, Bree? SNA, I'm sorry, sna.ipps.org. And again, this was a great year for our growers. If you would like to make a tax deductible donation to our endowment fund, you can find out more information on the website. Last, I want to invite you all to our Southern Region virtual happy hour and question box. The question box is always heated, but we always have a lot of fun and we learn a lot from each other. That's going to be October 28th at 6:30 p.m. Eastern time. It's so weird. And thank you, Bree, for hosting me. I'm traveling across the South right now, and I needed a safe home base. So we're having a lot of fun. So I think now, Cheryl, will go to our videos.
All right, we're going to play 3 student videos. They're real short. They're all less than 2 minutes long. So we will do that. Here's our first one. And here we go.
Hi, my name is Gabriel Campbell, and I am a graduate student at the University of Florida. For the past few years, I have attended the IPPS Southern Region conferences and have been involved with the student competitions.
Hold on, let me get the volume up. It's hiding from me. And I'm gonna start that over so you can hear it. Okay, here we go.
Hi, my name is Gabriel Campbell, and I am a graduate student at the University of Florida. For the past few years, I have attended the IPPS Southern Region conferences and have been involved with the student competitions. This was a very, very rewarding experience for me. It taught me how to communicate with industry professionals, other growers, and other researchers as well. And the meetings themselves have been spectacular. I've been able to travel throughout the South and see different growers and different technologies that they're using, and to communicate with other researchers as well. It's been a very rewarding experience for me, and I really urge any student interested in plant propagation or plant production to come to the meetings and be a part of the student competition. Thanks for your time.
All right. Hold on. We will switch maybe to the next one. This next one is Shay Keene. Make sure the volume is all the way up. And here we go.
Hello, my name is Shay Keene, and I'm a PhD student in environmental horticulture at the University of Florida. I first learned about IPPS back in the spring of 2016 when I was a brand new master's student at UF. With the encouragement of longtime IPPS member Dr. Sandy Wilson, I applied for the Vivian Monday Work Scholarship Program and was chosen as one of the recipients for the 2016 meeting in Virginia Beach. Working the registration desk was an excellent way to meet many of the members and learn about the opportunities offered through the organization, such as the Charlie Parkerson Student Research Competition and the International Exchange Program. By the end of the conference, I decided to become a member. In 2017, I attended the meeting in Dallas, Texas, and presented a poster as part of the Student Research Competition. In 2018, I was chosen as a finalist for the Student Research Competition and gave an oral presentation in Chattanooga, Tennessee, and won first place. Finally, last year, I achieved the goal I'd been pursuing since that first meeting in 2016, to be selected as the Southern Region representative for the International Exchange Program. In October of 2019, I spent 2 weeks in the south of England with the current international chair, Tim Lawrence Owen, and his wonderful wife, Annette, as my host. I toured nurseries and gardens and attended the European region's annual meeting in Stratford-upon-Avon, where I learned about the horticultural industry overseas and met horticultural professionals from across Europe. It was, without a doubt, one of the most incredible and impactful experiences of my entire life, and it would not have been possible without IPPS. I am so grateful for the organization and all the people I've met through it, and I look forward to being a member and giving back to the organization for many years to come. I hope you stay healthy and safe during these uncertain times, and I look forward to seeing y'all in Alabama next year. Thank you.
Well, I'm sorry that that's not very loud, but these are on YouTube, so we can send the links out if you'd like to review them. I think Judson's is louder, so let me just make sure it's as loud as it can possibly be. And here's some more Judson.
Hey guys, it's me, Judson LeCompte, and I wanted to share with you about my amazing experience that I had as an exchange recipient from IPPS European region. This is a collaboration between Southern and European region, and it was such a great opportunity. I was the first one to go. I got to go to over 20 different nurseries in England. I got to go to Cambridge Gardens. I got to go to Kew Gardens. I got to see amazing things. And guess what? It cost nothing. They used to chase me down when I was trying to buy a cup of coffee. So for our students and young professionals, this is a great opportunity for you to learn new skills, meet new people, and get a global perspective on our industry. So I was also fortunate to be able to host 2 of the European recipients. So once again, thank you for supporting the Education Foundation and this program. It has really changed my life and has gotten me to where I am today. So thank you so much.
Our international sponsors, Ball Horticulture and Harold's Fertilizer, and our North American sponsors, Bailey Nurseries, Bayer, ICL Osmocote, Nursery Supplies, Spring Meadow, and Star Roses and Plants. Your investment in IPPS is greatly valued and appreciated. Additional thanks to our Southern Region sponsors, Greenleaf Nursery Company, OHP, and Virginia Nursery and Landscape Association for your continued support.